Welcome everybody to this really exciting event, and I want to begin by thanking Jay and your colleagues in INSPIRE for all your efforts in many different domains, including getting this great symposium together this evening. I'm especially grateful to you because you've allowed me to duck out of an otherwise rather boring board meeting, this being much more fun. And however, I have to apologize because they're going to make me go back to that board meeting in a little while. And I only wish I could stay and listen to all the talks, which I know will be terrific. This has been a difficult year and a half, almost 2 years of COVID. I know everybody has worked incredibly hard and has stepped up in many different ways. But I've always believed that institutions have personalities and cultures that really are important at all times, but especially important when we're challenged. And I've been at Penn 25 years. I've stayed here because I like the culture and the people here. And I've never been more proud of my colleagues and all the people who I work with than over the past year and a half, when everybody has stepped up to do work to help each other above and beyond. We've always had a great culture of collaboration, of support for one another, but that's just never been more important than over this last difficult period of time. So I want to thank everybody for that. Our BGS students have continued to carry out really important experiments. It's actually been a great year in the School of Medicine. We've had more high impact publications than ever before. Grants are up, grant submissions are up, awards to trainees are up, and that despite all the challenges. And just in a month or so, we're going to be opening up a brand new hospital that you see looming over the Philadelphia skyline. Enormous efforts going into getting ready for that event. And at the same time, I know we're all proud of the many advances, including the mRNA vaccines developed here by Drew and Katy Carrico and their laboratories and colleagues. And that certainly makes me proud to see them getting the recognition they deserve, and more of that to come, I predict. So thanks to the speakers for being willing to contribute today. Jay, thanks again to you and your colleagues. Thanks for having me here to welcome everybody and sit back and enjoy what I know will be some terrific talks.
Hi everyone. I hope you guys can hear me. Again, thank you, Dr. Epstein, for such a wonderful introduction. Hello everyone tuning in. For those that do not know me, my name is Jay Ortiz, and I will be your moderator for today. I'm a doctoral candidate in immunology at Penn and the founder of the Penn Interdisciplinary Network for scientists promoting inclusion, retention, and equity, or Penn INSPIRE. Briefly, we are a group of students, postdocs, and faculty with the mission to improve access to education, opportunities, and initiatives for those communities that are historically excluded in STEM. As John mentioned, our forum today is centered around the COVID-19 pandemic, and we'll hear from 5 fantastic Penn scientists who have united to share the lessons they have learned during this unprecedented time and share their vision as to how to move forward. And so without further ado, I'd like to introduce our first speaker today, Dr. Susan Weiss, who is currently a professor and the vice chair of the Department of Microbiology, as well as the co-director for the Penn Center for Research on Coronaviruses and Other Emerging Pathogens at Penn Medicine. Her lab is focused on the biology of coronaviruses, including SARS, MERS, and SARS-CoV-2. So please join me in welcoming Dr. Weiss.
I'm really excited to be the first speaker for this session. And just in comparison to John, I've actually been here for now 41 years. I can hardly believe it. And I've been working on coronaviruses all that time as well. So I'm going to try to share my screen, which has been a little bit difficult here. Should that be the same way as we did it before, above my head? Oh, there we go. Okay. All right. I think we're here now. Okay. So I'm going to talk about, in this short talk, a very little bit about the history of coronaviruses, then something about the basic biology of coronaviruses, which will allow us to go into the other parts of the talk, which are to talk just a little bit about the origin of SARS-CoV-2, which has been a sort of hot topic lately, a controversial topic, and then a little bit about the emergence of SARS-CoV-2 variants and why some of these may be problematic or of concern, as they're called. So coronaviruses are they're RNA viruses. They're named, coronaviruses are a family within the nidovirus order, and they're named for their nested subgenomic mRNAs, messenger RNAs generated during infection, with nido meaning nested. And these little spiky things protruding from the virus particle are what give it this crown-like morphology, and hence the name corona. On the right is a schematic of the coronavirus virion. Without going into a lot of detail, there's a very, very long RNA genome encapsulated inside of it, complexed with a nucleocapsid protein. And on the surface, there are three viral proteins that are inserted into this blue membrane that's derived from the host cell when the virus is assembled. I'm just going to highlight the spike protein, which I think everybody's heard about by now, and it's really important because it's what attaches the virus to its receptor on the host cell surface. It mediates viral entry, and it's the target for which the mRNA vaccines, against which the mRNA vaccines are directed. They have a spike protein sequence in the mRNA in the vaccine. So all the antibodies are made against the spike protein. There are just a few other proteins in the viral membrane called membrane and small membrane protein. A little bit of history here. I can't really animate because I'm not on. Can you see if I do this? Can you see it now as a?
No, it's still on the normal.
Oh, okay, because I can't really animate them, but okay, I'll try without animating. Okay, so coronaviruses date back to the earliest in the literature in the 60s and 70s. There is these two cold viruses called OC43 and 229E, as I said, causing the common cold. And from that time until about 2002, during the first SARS-CoV epidemic, there was a lot of basic biology done on these viruses. We understood what the proteins are. We knew that the spike protein was what attached the virus to the cell. The spike protein receptor was identified. A lot of this work was done in mouse hepatitis virus, a mouse coronavirus that's been used as a model for much of the molecular biology that's known about these viruses. In 2002, SARS-CoV-2 emerged in southern China and caused this severe acute respiratory disease. And this was the first notion that these coronaviruses could cause lethal or pathogenic, really pathogenic disease in humans. And that's why I labeled this the pathogenic human coronavirus era. And after SARS, people looked for more coronaviruses, and HKU1 and NL63 were identified about 2002, 2003. They caused pneumonia and bronchiolitis, respectively. And then things were kind of quiet until 2012, when Middle East Respiratory Syndrome coronavirus, or MERS coronavirus, emerged in Saudi Arabia, causing a really similar disease to SARS. Although I'll show you in a moment that the epidemics were really quite different. And then things again were quiet until the end of 2019, when SARS-CoV-2 emerged in a different part of China, in Wuhan, causing the epidemic that we've all been living with, or the pandemic, I should say. And just to highlight, all of these viruses are believed to have origins in animals. They're all zoonotic, so to speak. So SARS-CoV-1 came from a bat, which was transmitted to a civet and then to humans in 2002. And then MERS also came from bats, a different bat from SARS. It was transmitted to a camel, and it has a huge reservoir in camels, and then to humans. And SARS-CoV-2, which I'll talk about a little bit more in a moment, I'm almost completely convinced it had a bad origin. There may be an intermediate species, and of course it transmitted to humans in 2019. But I want to point out that these other non-lethal viruses also had origins in other animals. OC43 was believed to originally been in a rodent and then transmitted to cattle or pigs and then to humans in the late 19th century. And 229E also has a bad origin, transmitted to camels like MERS, and then to humans again in the late 18th century. And NL63 is believed also to have been transmitted from a bat to a human in the 13th to 15th century. So it's maybe not so surprising that these pandemic viruses came from other animals. So I just want a little bit more about these epidemics. So SARS has its origins in a horseshoe bat transmitted to the civet cat, as it's called, which were used for food and in China at the time, and were found in these wet markets, and then transmitted somehow to a human. But the interesting thing about that epidemic is it only lasted 8 months, and then it was gone. There were about 8,000 infections with a higher mortality rate than SARS-2, but a lot fewer people were infected, and almost all of it was in China and Hong Kong. MERS, in contrast, also similarly came from a different bat, but it has a very large reservoir in camels, both in Saudi Arabia and Africa. But only in Saudi Arabia is it transmitted to humans. There's limited human-to-human spread, but it continues to infect humans. There are still new cases in 2021. Not a huge number of cases considering how long it's been around. So these two viruses are very similar, cause similar diseases, but their epidemics or the epidemiology was quite different. So then we get to SARS-CoV-2 that I'm pretty sure came from a bat. We don't know an intermediate species. It was then transmitted to humans, and then with lots and lots of human-to-human spread, as we know, with millions of deaths and millions in the world, and hundreds of thousands in the United States, and really, as we all know, it's just been a pretty awful experience. So I want to talk just a little bit about the coronavirus, about the biology of coronaviruses, so we can look a little bit more into the origins and also into where these variants come from. So just quickly, these are the genomes of all the human coronaviruses. They fall into different genera. There are 3 lineages of beta coronaviruses that include the OC43HKU1, SARS, and SARS-CoV-2 are these so-called sarbecovirus, and MERS is in yet another lineage. Alpha coronaviruses include these 2290 and L63. All of these viruses have similar structures. Their genomes, 16 replicase proteins. These are conserved proteins and really good drug targets, which I'm not going to have time to talk about. Then the spike E, M, and N proteins are all arrayed in the same order. In this part of the genome, the spike, of course, is the vaccine target. And I'll talk about that in a moment. And then these proteins, accessory proteins, are important for combating host cell responses. Just really quickly, coronaviruses, the life cycle, the spike protein attaches to its receptor. The virus and the host membranes then fuse, and the viral RNA is transcribed into messenger RNAs, and also the genome RNA is replicated. The nucleocapsid protein combines with the new genome RNA, and it buds into these in a compartment between the ER and the Golgi. And then we see new virus particles in vesicles, and they're extruded from, or they're released from the cell into the extracellular space. And spike protein is also put on the cell surface, and I'll show you later, these proteins can cause fusion of infected cells, and that's important for virus spread. So, and here's the spike protein, a diagram. The spike protein has 2 subunits called S1 and S2, and during viral entry, they have to be cleaved or cut in half by an enzyme called furin. And some viral proteins have what's called a furin cleavage site, and that's shown here. It's just a bunch of basic amino acids. And that becomes important in talking about the origin of the virus. So just remember that the spike S1 has the region that binds to the host cell receptor. It has a cleavage site, and then there's this S2 subunit. Okay, so there have been all kinds of alternative theories about where SARS came from. And why? Because a close ancestor in a bat or an intermediate species have not been identified yet. So that makes people suspicious. This is the only known sarbecovirus or MERS-like or SARS-like virus with a so-called furin site for cleaving S1 and S2. And some people think that suggests it was engineered by humans. I don't believe that. And the labs in Wuhan hunt viruses in bat caves and bring them back to the lab. So some people claim this could be a so-called lab escape or lab-acquired infection. But I think that's not true, and for the following reasons. I think there's no way that it was engineered by humans, and it's unlikely to be from a lab. Because very similar viruses are found in bats. They're not exactly the precursor, but there are viruses in bats that can infect humans directly. This virus does not resemble any known recombinant viruses and had never been seen before in the Wuhan labs. And also many other natural coronaviruses, including MERS, have these so-called furin sites for cleaving their spike. And the most compelling reason is that it's really not possible for anyone to know how to design a virus with the properties of SARS. That is, a virus that can spread pre-symptomatically and infect as many people as it does. There's no way to know how to do that by a human. It has to be by natural selection. Okay, so now that we've talked about that, I just want to talk a little bit about the variants and where they come from and how they're generated. So here's the viral genome RNA, and when it's replicated, the genome RNA is translated into a bunch of proteins that replicate that genome. They make this anti-genome, so to speak, or the negative strand, and those are copied back into new genomes. And every time a virus with a long genome like this, 30 kilobases, replicates, it makes at least one mistake or one amino acid substitution. And if that happens to be in the spike gene, most likely, and it happens to give the virus an advantage for replication or spread, it will be this genome will be selected and will kind of take over, sort of like some of the variants that we've been hearing about. So let's look at the variants. I wanted to show this animatedly. It's much easier to look at, but here's the alpha or the UK variant, and it's got these in red, these amino acid substitutions or mutations in the spike shown here. The beta or South Africa derived virus has these green ones here. And the Delta virus, the one that really pretty much every clinical isolate here at Hup and Shop pretty much today is a Delta virus. It's different. It has all of these amino acid mutations. So why are these variants of concern? Why is everybody so worried about them? Well, there are, I think, at least three different reasons. Some of them cause increased receptor binding, like this N501Y mutation, which is pretty common. This mutation just allows the spike to bind better to its ACE2 receptor. Immune escape, so this E484K mutation here is in an important epitope for antibodies to attach to. So mutation in that epitope might make an antibody less likely to recognize the spike protein. And then the one I think is really interesting is this right here in the cleavage site, this P681, either R or H mutation, makes this cleavage signal more basic, and the more basic the cleavage signal with histidine or arginine, the more likely it is to be cleaved, and the more likely it is to be cleaved, the more well the virus, it makes the virus more able or fuse into the cell better, and also to fuse cell to cell and spread better, and I'll show you in a minute what I'm talking about. And then mutations in other genes may also contribute as well. So any of these mutations that are selected by just natural selection to make the virus replicate better, and that is of concern. I don't think they necessarily make them immune escape, but they do make them spread more quickly. So just to show a little bit more what I mean. So here's a vaccinated individual is vaccinated with against the spike protein. So these antibodies here that are made are all going to recognize spike protein and prevent the virus from infecting the cell. And so these antibodies are going to be directed all against the spike protein, but it's really only the ones, say, against this E484, which may be less likely to be able to be active against the virus that has this mutation. So that's why there's a worry about a mutation like that in the spike protein. And then the other thing, so variants that have this fusion site mutation, this H681P or H, are going to make, this is what cell fusion looks like. So this is just nuclei of cells that are uninfected. These are infected cells. The green shows viral protein, and you can see that there are these big masses of fused cells, all these nuclei together. And this is caused by the viral spike protein on the outside of the cell causing fusion with the next cell. And the more basic this cleavage site, the more you're going to get this fusion. And this is actually stem cell-derived cardiomyocytes, and these are just tissue culture cells. They're not really they're cell lines. But this is cardiomyocytes from stem cells, and you can see that they also are fused by SARS infection. So this is another reason why a variant might spread better. So just a few questions to think about going forward. Will variants continue to emerge, or will they reach an optimal fitness and stop? So I think it could be that at some point this virus can't tolerate any more mutations and it will have to stop making them. I don't know. I just think that's possible. Are vaccine breakthroughs more common in Delta variant infections? I think it's hard to know because all the virus we see now is Delta. Will we get vaccine-resistant variants? I don't think we'll get completely resistant vaccines. I think they may be against one epitope, but there are many epitopes to which the vaccines are directed. Will SARS-2 attenuate as we believe for other previous common coronaviruses? I think that's possible. Like I showed you in the beginning, OC43 started as an epidemic, a pandemic, and I think perhaps some the other ones did as well. So it's possible that as the virus evolves, that it could attenuate, and we're kind of immunized anyway, so it might kind of, I think it could become sort of just a virus that lives with us. And then the other question is how to prepare for future emergent coronaviruses, and I think the way to do that is really to try to study them, to understand them, to look in animal species and try to figure out where they are and where they may emerge from. And also to develop more antivirals that can be effective against all the coronaviruses, as well as vaccines. And I sort of said this: develop pan-coronavirus antivirals, vaccines, identify and characterize coronaviruses and other viruses as well from bats and other species, and support basic virology research, because only by understanding these viruses and how they replicate can we understand how to try to combat them. And then finally, I know I didn't really talk about any work from my lab, but I do want to acknowledge my lab that's been working really hard during this time, really during the whole pandemic, and during the hours that I give seminars, they're working away. So thank you. I'll stop here. Okay.
Okay, we're just waiting to bring Jay on screen.
Sorry, my computer is not used to so much excitement, right? Thank you, Dr. Weiss, for such a wonderful talk. Let's see, we don't have any questions right now, but maybe I'll start throwing off some questions that maybe have been on my mind, and sort of maybe just to get to the general audience that we have here today. So for example, we keep hearing about how these novel variants are sort of this collection of mutations, right, that enable the virus to spread more efficiently, to fuse with other cells, right? Someone might ask, is it just going to keep adding mutations? Like, is a combination of these mutations might be harmful to the virus, right? And how does that sort of like, well, as they're, yeah, go ahead.
Okay, so only the mutations that are beneficial to the virus are going to survive. The other ones will just be drowned out by the more effective ones. And remember, the virus wants to spread. It doesn't want to kill people necessarily. So the selection is really for spread, not for pathogenesis. So I think that it can get more mutations, but at some point the proteins are not going to. There's some how do I put it? There's some restriction on their structure and stuff like that, so they can't change too much. So at some point, I think they'll stop. I personally think they'll stop changing, and I don't think they're going to get more pathogenic. They may get less pathogenic because it may spread, but not be as virulent.
We have one question from the audience that says, "Thank you, Dr. Weiss. Is there a concern that the spike protein mutation will evade the popular rapid antigen tests?"
I personally don't know. I doubt it. I don't know what they're looking at the antigen test, but if they're looking at more than one protein or more than one epitope, I don't think that would happen. I doubt these tests are focused on just one small region of one protein. That would be a mistake for that kind of test. So if it's looking at a broader thing, like I was showing before, that the vaccine, for example, will elicit many different antibodies and T cells and things like that. So if it's testing for an array of antibodies, I think it won't evade the rapid test. I can actually see the questions too.
Thank you.
Antibodies to the spike protein.
Oh, nice.
Block the cell-to-cell fusion that is mediated by the spike protein. So I guess it would depend on which antibodies and what. I don't know the exact structure of the spike protein on the cell surface compared to on the viral surface, but they could. I think they could.
All right. I think with that, just to kind of keep moving along, really thank you so much, Dr. Weiss, for your time and for this wonderful talk. All right. So our second speaker today is going to be Dr. César de la Fuente. He is currently a presidential assistant professor at Penn, where he leads the machine biology group, whose goal is to combine the power of machines and biology to understand, prevent, and treat infectious diseases. And he's going to talk about how his team recently developed this miniaturized portable version of a rapid COVID-19 test, which is compatible with smart devices and can detect SARS-CoV-2 within 4 minutes with almost 100% accuracy, right? So please join me in welcoming Dr. de la Fuente. Thank you so much.
Great, thank you, Jay. Can you hear me okay?
We can hear you great. Thank you.
Okay, perfect. Yeah, I just wanted to thank Jay and the organizers and for inspiring us all through through the organization, bringing together people now to discuss COVID-19 within this context and bring really a diverse set of interested parties to listen to this conversation. So I thought I would present a little bit some of the work that we've been doing. Let me just share the screen here. And okay, so I'm going to briefly tell you about several of the prototypes that we've developed in the lab to try to detect SARS-CoV-2. From the very onset of the pandemic, my lab, we felt a responsibility to try to contribute something. We were at the time, before the pandemic, we're working on developing diagnostic technologies for bacterial infections. And when the pandemic hit, we repurposed some of the stuff that we were doing to try to diagnose COVID. And we identified early on two things that we wanted to focus on to try to do something about. One of them was we wanted to develop technologies that were inexpensive, so that anybody could afford them. Not only people with economic means, but perhaps people that live in low resource settings or in developing countries or in communities that really need it the most. So we really thought about that quite hard as a lab. And the other aspect that we really focused on was we wanted to develop a technology that was that allowed a rapid detection of the virus. And this is because there's a lot of evidence already that if you can achieve rapid detection, this is an excellent tool to be able to prevent the spread of the disease. And of course, if you prevent the spread, you can prevent outbreaks, you can prevent hospitalizations, and you can hopefully prevent a lot of deaths. And so, really, that was the concept. Can we develop something that anybody could use potentially? And then this is basically just the background, the context, right? We live in a world that is highly inequitable, and that has only heightened during the COVID-19 pandemic, where we're seeing an incredible inequity in terms of distribution of vaccines. The last time I remember reading, it was basically 10 countries in the world acquired 75% of all vaccine shots. And in many countries, the percentage of individuals vaccinated is less than 10%. And I think it's important to think about this and think about how we can help, perhaps, or at least work towards disrupting these health inequities that are present in our society. But anyways, going back to the science, so we focus on developing, or so far we have developed 4 prototypes that I'm outlining in this slide. The first one was rapid, the second one was lead, and the third one was color. So I'm going to briefly tell you about each of them. So let's start with the first one, rapid. So this technology uses electrochemistry. Basically, it allows you to detect the binding between the virus, more specifically the spike protein, which is on the surface of the virus, the binding of it to a receptor, and this technology captures that binding affinity in a very precise way. And these are some of the statistics of this, or the parameters of this test. The low cost, we definitely achieved that with this first prototype. It's actually $4.67 per test, and it's actually compatible both with the laptop. You can connect it and you can get your testing done on a laptop, or it's also compatible with a cell phone through a USB drive machine. You can connect your test, which is this strip here, and get your results using an app. But I'll tell you more specifically. So we can use saliva samples or nasopharyngeal samples. This is essentially how we build it. We have an electrode, which is essentially a chip. We functionalize onto the electrode ACE2, which, as you've heard from Dr. Weiss, it's the natural receptor in our own bodies, in our own cells, for the virus, in such a way that whenever the virus invades our bodies, it's going to bind to ACE2 on our cells, and that's how the infection process begins. And so we're basically recapitulating here, or mimicking what is happening in nature, right? The virus approaches our cells and binds to ACE2, and that's how it gets into the cell, right? So here we're basically doing the same thing. We're putting the ACE2 onto the electrode, and then when we have a sample that contains the virus is going to bind to ACE2, and we're going to be able to detect that binding. These are some of the limit of detection parameters or results that we obtain in different media. And I don't need to get into all the details, but basically it's highly sensitive in terms of analytical sensitivity, meaning we can detect really minute amounts of the virus on specific samples, saliva or oropharyngeal samples. More specifically, this is how we actually do the engineering, how we build this test, this diagnostic. So we have the electrode, then we put this reagent here, which allows us to anchor something else. And as something else, we use ACE2. We use a recombinant version of ACE2 that we then anchor onto this platform. What we then use is we use bovine serum albumin. Basically, this brings stability to the whole thing. And then we use Nafion 1%. It's basically a compound or a molecule that allows us to prevent anything else from filtering into the test that may confound the results. And again, we electrochemically validate each of those steps to make sure everything is right. And we've done substantial studies on this, but one of the things that we do is we test for cross-reactivity to make sure the test is detecting SARS-CoV-2 and not other viruses. And we've tested a number of viruses here. And then this is how this test, Rapid 1.0, the first prototype, how it compares in terms of the cost per test and the detection time with respect to existing methods that are FDA approved today. And in terms of clinical performance, this is the data when we take COVID or non-COVID samples from the hospital and we assess them using Rapid. This is the sensitivity, specificity, and accuracy data for nasopharyngeal and oropharyngeal samples, and also saliva samples. So the test is not perfect in terms of accuracy, but it's around 90%. And then we're also able to detect some of the variants. We've detected consistently the UK variant, as Dr. Weiss mentioned. A lot of these variants actually. Funny enough, they bind more strongly to ACE2. So we actually pick up through our test that increased binding affinity with a stronger signal. So that was actually an interesting thing that we found. Okay, so moving on to the second test, we call it LED, low-cost electrochemical advanced diagnostic. The interesting thing here is that we wanted to use materials that were highly accessible to be able to build a test, kind of like a do-it-yourself sort of test. And basically, what we did is we took we decided to test whether we could use pencil leads. And I have here one of the prototypes. Again, pencil leads, like what you see in the office and around you all the time. And pencil leads, the interesting thing they have is that they're electroconductive. So what we decided to do is we decided to build these plastic tubes, have these 3 pencil leads. One of them we functionalize with ACE2. And then you can close it. You can have a sample here. Again, it can be saliva or nasopharyngeal sample. You can apply an electrical current onto the pencil leads, and then you can do the detection in that way. Again, the detection time is delineated there on the slide, is about 6.5 minutes, and the cost per test is $1.50. But again, the concept here is that we wanted something that was highly accessible, right? A material that anybody has access to, anybody can buy, and that's of course pencil leads, and that's how we did it. So this is what the prototype looks like. Like I showed you, it's basically a plastic tube with the 3 pencil leads, and that's how we do the detection. We use a slightly different technique here, square wave voltammetry. I won't go into all the details, but it's also part of the realm of electrochemistry. Again, we ran cross-reactivity studies to make sure that the signal is specific to SARS-CoV-2. And here's what I was telling you, that in the case of the UK variant, the signal is even more pronounced. Again, with this particular technique, the signal is captured by a decrease in this curve. So the more decrease you have, the more signal, the more positive detection you have. And we can see that it doesn't, there's no cross-reactivity with H1N1 or other viruses. And again, the price to detection time matrix of lead compared to existing methods, it really falls into a category there that is highly desirable from a testing standpoint. And here's the clinical performance using nasopharyngeal swabs or saliva samples. And here's the total number of samples in each case, and sensitivity, specificity, and accuracy for both subsets of clinical samples. Okay, and finally, I'd like to tell you briefly about color. This is the 3rd prototype that we developed. This is slightly different. It doesn't use electrochemistry. It uses nanotechnology, and it's I think of it like a pregnancy test, but for COVID, in the sense that you get your result through a color shift. The detection time is less than 5 minutes. The cost here is this is really interesting. It's 15 cents per test. And I think this can be really potentially important in the future. This is how we do it. We use basically nanotechnology. We take advantage of the plasmonic effect of nanoparticles, and again we use ACE2 as the binder. The limit of detection is it's delineated here. It's not this method is not as sensitive as the electrochemical methods that I've talked about before, but it's still sensitive enough, in my opinion, to do massive mass testing. The other interesting thing is that, so basically the way it works is you have a cotton swab. We functionalize the cotton swab with ACE2. Then we use nanoparticle technology to do, through a plasmonic effect, you can see a color shift within minutes if the virus is present on a particular sample. And because it's a color shift that you see qualitatively, you can also quantitate this information using we've been using actually a free app that quantitates pixel intensity, and we can do it in that way. So that through the app you can actually see, okay, is this significant or not? Is the signal that I'm getting significant? The signal is also correlates very nicely with CT values that are typically yielded in qPCR results, which is the gold standard, right, for COVID testing. And so we can detect high viral load samples, medium viral load, and low viral load samples, and the correlation is quite nice between between color and PCR. Again, we ran cross-reactivity assays to make sure it's selective, the method towards SARS-CoV-2, and it was. And then also reproducibility assays to make sure that when we manufacture different batches of the swabs, the cotton swabs of color, that the results are the same. And then this is the clinical performance using COVID's clinical samples. Again, these are nasopharyngeal swabs, And here's the sensitivity, specificity, and accuracy out of 100 total samples that we tested in this case. That's all I had today. So it's a brief sort of a summary of the different prototypes that we've been able to develop. And finally, I'd like to thank the lab members, the team. It's really a privilege to work with them every single day with incredibly smart and highly, interdisciplinary actually, and diverse people. They come from all sorts of backgrounds, including nanosciences, microbiology, computer science, physics, and so it's really a pleasure to work with them every single day. Thanks to our funders that allow us to do the work we do, and thanks. And here I'm just leaving a link to our website, my email address, and Twitter account in case anybody wants to get in touch. I'm always available. So thank you very much for your attention. I'd love to take any questions.
Hi, Dr. De La Fuente. Thank you for that wonderful talk. This is incredible technology that you guys have sort of started using. I really feel like, I know that the specific tests that you have done, are specific to SARS-CoV-2, but it seems like this could be really applicable to other common viruses and other potential emerging infectious diseases, right? Have you guys started working on, adapting all these very economical tests to the detection of different, emerging pandemics, for example?
Yeah, Jay, that's a great question. So we're actually we're applying it to detect other viruses like herpes virus, for example. Also bacteria. Super bacteria are a huge problem. They're becoming increasingly resistant to all antibiotics that we have available. So we're also interested in trying to detect them early, bacterial infections. But conceptually, some of these technologies could be applied to detect other things like biomarkers. As long as you have something that binds to something else, and both elements are proteins, which we're very familiar as to how to manipulate them, how to work with them. I think at least hypothetically or conceptually, we should be able to, through this electrochemical technology, at least detect that binding between two different elements. And so I think the applications can be potentially quite broad. We're mostly focusing on infectious diseases, so viruses and bacteria. But I agree with you. I think the future is quite promising and exciting.
Okay, so there is one question in the chat from Alyssa. So they're asking, where do people with severe illnesses fit in the equation? They're not eligible for the vaccine, but are still at risk because unvaccinated and unvaccinated people are able to contract the virus.
Eligible for the vaccine, but are still at risk. Yeah, I mean, it's an interesting question. I'm not sure if we're talking about people with severe illness that are unvaccinated or whether they're vaccinated. But I mean, as far as I know from breakthrough infections, it's a minority. As far as I know, I'm not an expert in this, but the likelihood is not incredibly high of breakthrough infections, which means when you're vaccinated and you still contract COVID. And I also, from my understanding, the people that have breakthrough infections, those infections are not as severe as if you're unvaccinated. And so I think that really speaks to the value of vaccines and getting vaccinated, and that the value that provides both individually and as from a population standpoint, right, in a group. Not sure. I don't know if that answered the question. Sorry if it didn't.
Awesome. Thank you so much, De La Fuente, for this excellent talk. We will now move on to our third speaker today, which is Dr. Allison N. Butenheim. She is currently an associate professor and the Silverstein Endowed Term Chair in Global Women Health at Penn Nursing. And so using insights from behavioral economics and related disciplines, Dr. Brittheim designs and trials interventions to change behaviors that are central to infectious disease prevention. She recently co-founded Dear Pandemic, which is a collective of interdisciplinary women scientists that are addressing science communication during COVID-19 with accurate, timely, and curated science, news, and lifestyle, as well as guidance. Please join me in welcoming Dr. Brittheim. And sorry if I chopped your last name.
That's okay. When your last name is Buttenheim, you get used to lots of different ways of getting it pronounced. Are my slides showing? Are my slides up?
Yes, they're all great.
Perfect. So hi, everybody. This has been a really interesting forum. I learned a ton from the last two talks, which was wonderful. I'm a behavioral scientist by training, and so getting to learn some of the bench science and the diagnostic science is great. But I'm going to talk to you today about a different pandemic we're having, the infodemic, related to COVID-19, and share some work we've done on an information campaign called Dear Pandemic, which is mostly completely separate from my day job being a professor at Penn Nursing, and I'll tell you some of that story. I have found it helpful in the many, many talks about Dear Pandemic that I've given over the past bit. If people feel free to go check us out while I'm talking, I don't mind at all if your attention is somewhat split. But we're on Facebook at both Dear Pandemic and Querida Pandemia, our Spanish language site. We're on Instagram, Twitter. We have a website. We're also on LinkedIn, if that's your jam. So please do feel free to, if you have no idea, I've never heard of us before, and you're wondering what I'm talking about, find a double screen me. Don't mind that at all. I'm also, and I'm a little nervous about this, going to attempt a poll everywhere poll, which you can get to really easily at either the pollev.com with the / and my name and the number 510, as you can see at the top of the screen, or just text my name and 510 to 22333, and hopefully this poll will pop up. I'm just curious if kind of read the room in terms of where people's familiarity is with Dear Pandemic. Either you already follow us on one of our channels, maybe you've heard of Dear Pandemic, but don't follow, or like you've never heard of it before right now. So I'll give people a couple minutes either to go find us if you want to check us out, or maybe attempt to respond to the poll. I've been using Poll Everywhere in my class this fall at Penn, and it's kind of fun. I'm seeing a couple responses start to trickle in. Maybe when we get to 15 or so, I'll reveal the answers. And let me just start talking about where Dear Pandemic came from. So in late February 2020, turn your mind back, I and my colleague from grad school, Malia Jones, who's at the University of Wisconsin, we're both fielding a lot of questions with friends and family about, do we have to pay attention to this? Do we have to cancel travel? What's going to happen? Should we worry about this? And we're answering those questions kind of one-off. Malia wrote an email that got widely circulated. It was a Google Doc and got shared actually about 100,000 times. And my colleague here at Penn, Ashley Ritter, said, your info is so great and so many people need it. Would you consider, having an Instagram account or something? And I consulted with Malia. She said, sure, that sounds great, but I've got to go be on Dr. Phil next week, and I need to focus on that because somehow he's heard of the info I've been sharing. Go ahead and get it started, and I'll see you there. So on March 10th, I started an Instagram account called Those Nerdy Girls, and that was based on something that a mutual friend of Malia's and mine on Facebook had said. He's like, I'm just going to listen to Those Nerdy Girls for the rest of the pandemic. I don't know what else to do. And a few days later, in consultation with my family, who loved the Nerdy Girls handle, but said the project needed a different name, my husband actually suggested Dear Pandemic, kind of like Dear Abby. So sort of an advice column for the pandemic. That went live on March 13th. Let's see how the responses are going. So great, awesome. We have a very Dear Pandemic naive audience, which is fantastic. At least for the N = 16 people or so who responded. Thank you for engaging. So here's Malia and me. Here's Ashley saying you should get this on social media. Again, March 13th, we're on Facebook as Dear Pandemic and Instagram. We realized we needed help. Within a couple of weeks, we had recruited about a dozen colleagues, all female PhD prepared scientists in the social science and clinical science. Many of these folks are nurses. And a few months later, that group had actually grown to over 30, many with Penn connections that I've indicated there. Scientists of all stripes, and several wonderful volunteers and student interns. Almost everybody completely volunteer, a couple of exceptions that I can talk about. And if you're interested in more about the origin story of Dear Pandemic, we've had some really lovely profiles over the past year and a half, most notably in National Geographic, Bajillian Kramer in March 2021, but in Fortune, locally in Philadelphia Citizen, and many of the schools where we trained or work have also done nice profiles like this University of Chicago one on Dr. Lindsay Leininger. So there's lots of Dear Pandemic lore out there if you're curious. So what is it we do if you've never heard of us? Basically once or twice a day, someone on the editorial team writes a brief post anywhere from 100 to 500 words about some sort of question related to the pandemic, really broadly defined. It could be a clinical question, a science, a diagnostics, a therapeutic, a mental health, a wellness. That over the course of the next 48 hours, it gets archived on our website and tagged for easy searching. Our Instagram team migrates it over to Instagram with a few tweaks. And a selected number also get turned into tweet threads on our Twitter account. A selected group of those gets translated into Spanish and put on Querida Pandemia. Querida Pandemia also has its own content, and some of it goes to LinkedIn as well. So that's basically this sort of what we do. We also do a lot of other things. We manage the project on a very active Slack workspace with lots of other things going on behind the scenes. We've forged many partnerships of different types. Since March 2020, 2 examples there with Wisconsin Head Start and Impact. We've secured some small grant funding from RWJ, from the Department of State for various activities. For about 40 weeks in a row, Malia hosted a Saturday morning live Facebook show with either Nerdy Girls or some outside guests. We've started writing scholarly manuscripts and publishing those about the project. And we do things like infographics and other SciComm, science communication work. This was our smart infographic last summer that's actually really had some good staying power. We also do a ton of media interviews. Those started coming to us individually as scientists, and I think the Nerdy Girl brand kind of built such that, and you can see this on our website, we have a whole section called Nerdy Girls in the News. We've done an average of more than one media placement per day since last February or March. That's everything from the New York Times or the Washington Post, calling one of us, to a lot of local publications. We're in Wisconsin and New York and London and Philadelphia, all publications of all kinds. So sort of our voice and our brand are out there. But what I want to talk today about is sort of tackling the infodemic, right? Tackling misinformation and disinformation. And I want to situate that in where we see Dear Pandemic's impact. And from day one, we're all scientists, we all do a lot of evaluation of different kinds. We've been thinking very strategically about our impact and saw sort of 5 domains where we might make a difference. One, of course, around changing behavior. Like we wanted people to not travel back in late February, early March. We want people to wear masks and et cetera, all the different behavior change. We also really want to build people's ability to assess risk to inform decisions. We can sort of give a man a fish in terms of telling someone whether a certain situation is risky, but what we'd really like to do is teach that man to fish, or women, usually most of our followers are female, by helping them think through sort of risky decisions. There was just a lot early on about navigating the pandemic. Think back to like, do I wipe down my groceries with Clorox wipes? Tons of mental health impacts that we were really wanting to be astute and helpful in. And then also hoping to kind of spread the nerd around, you too can follow the emerging science and policy and become very literate in that. So we've been thinking a lot about evaluation models and how to do that with the data that we have access to and how we might collect data. But starting from, the middle of last summer, because I was still talking 2020 here, 2 additional areas of impact really emerged as being very, very important to what we were doing. One was improving information hygiene, right? How people are sort of taking in and using and spreading information. And relatedly, how they're navigating the infodemic, which is, this sort of fire hose of info that is coming to us since the pandemic started from all sources, and figuring out sort of what to do about that. So another way of looking at that is, the scope of our impact has evolved from thinking about and wanting to build literacy and self-efficacy for the kind of information that's shown on the left here, right? How disease spreads. And I don't know if you remember this image that circulated early on. This got a lot of traction on our site when we reposted it. Evolving to also be about the image on the right? How information spreads and what we can do or not do to sort of nip bad information in the bud. So let me sort of zoom in on what we do specifically and how we think about Dear Pandemic as a platform for combating disinformation and misinformation. And I could speak like literally for 2 hours about this, but I will just focus on sort of 2 elements that we think are really important. The first one is trust, and trust really turns out to be the engine for us being able to drive our misinformation and disinformation efforts, countering those, I should say. When we started, we were about content. We're like, we need to get content out there. And we realized that content sort of drove reach. As we wrote good stuff that people were interested in, more people sort of spread it. And we were slower to realize how much trust was also a gear, a cog in that gear, or a gear in that engine. And the way we know that is that people mention it. Our followers are very active in commenting on our posts. We have a very active community. It's very responsive to each other. And we can really see in those comments, and these are all from our Facebook page, how important trust is. I won't read through all of them, but you can see that the word trust pops up in all of them in different ways, really confirming for us how important it is to our community that they trust the information that we post. We also know that they trust us because in the sort of social media world, shares are kind of the currency. So again, this is a Facebook metric. We can have comparable ones on Twitter and Instagram. But here's a post. It was a very popular post. I think it was our most popular post at the time. I think it's since been beaten. But it was when a lot of adults were vaccinated, but no kids were, and there was a lot of questions about what to do. Like, what can we do now as this sort of mixed family? And that post alone got almost 4,000 shares. So we're now a community of about 170,000 followers across our different platforms. In a given month, we reach between 1 million and 6 million people, depending on how much of this sort of sharing there is. This post alone, you can see, reached about 400,000 people. But the fact that almost 4,000 people shared it is also to us sort of an endorsement that we are a trusted source. That's not true on every social media platform, but on Facebook, a share really is often an endorsement. And we pay a lot of attention to the text that people post when they share our posts. So the little header that they'll put on below, above what they then repost from us. And those are fascinating. Often it's just like, hey, everybody in whatever page or community I'm posting this to, like, you should really read this. Or like, finally, a great description of, excess mortality or whatever it is. So yeah, just we are thinking a lot about where trust comes in all of this. The second element I want to talk about is how we listen to our community's concerns. We're an editorial endeavor, right? We're a content provider. And so it's really important to us as we select topics to write about every day that they are responsive to the questions people have. We realized early on that it was very hard to manage that just by sort of scanning the comments within a post, because a post about wiping your groceries down with Clorox wipes might have embedded in it a question about, the vaccine phase 3 trials. And so we put just a question box on our website, and we have some amazing sort of volunteer community engagement folks who will go through the Facebook page and the comments and encourage people to just click right over to the question box and put some questions in there. And that a synthesis of that question box every week is our starting point for putting together our editorial calendar. So that's a pretty manual, pretty labor-intensive version of what we could call social listening, right, or user engagement. And so we are looking for opportunities to sort of scale that. We are one of many, many kind of social listening endeavors that are going on. But what it helps us do is then build that into our evaluation of like, are we answering the questions our community has? So these are all posts that were very much in response to questions that our followers or our community had. And when we do, sort of post-level evaluation, which is something we're trying to get to, one of the things we look for as a variable is this a question that came in the question box? We're also pretty proactive in sort of coaching our community around infodemiology and misinformation. So we actually don't get a ton of inbound questions about that. They usually take the form of, my crazy uncle on the left here, and we can respond to those. But we will also take the initiative to just write up like a quick, how-to for debunking dangerous and false vaccine rumors, or some sort of staged, really easy to consume approaches to, for example, fighting fake news. Where this is going for us as scientists is into the sort of natural language processing, machine learning, AI type area, which is new to me. Like my science is designing behavioral interventions and then evaluating if they work. What you're looking at, and I realize it's busy, but I think the sort of entirety of it is kind of compelling, is an analysis of all 3,000 questions that we've gotten from the question box since it opened last August, so about a year's worth of questions. And each word cloud is one topic generated through natural language processing, which sort of says, here's a co-occurrence of words or terms that tend to hang together in a coherent way, and any given question can be sort of tagged as more likely or less likely to be under that topic. I hope that explanation made sense. And the relative size of the words within each word cloud is, the most common words that sort of identify that topic. So, we're really interested in this, both in the types of questions that have come up, the change in those prevalences or frequencies of the topics over time, but also just kind of the typology of them. So let me just highlight a couple of examples of what I mean by that. So here are the 5 topics under the broader vaccine topic that are related to the vaccine, but you can see they're actually pretty different from each other. So the top right is this sort of, when we're fully vaccinated, or I'm fully vaccinated but my child isn't, how do we handle that? What do we do about sort of being indoors or being in a household? That's pretty different from the one right below it, which is really this topic kind of peaked as the phase 3 trials were coming out and then the vaccines were rolled out. When do I get my first dose? When do I get my second dose? Does it matter if I get Pfizer or Moderna? How many weeks should I wait? Are boosters coming? Right, a really different question from this sort of family management question. So that sort of discernment of topics is of interest to us. And then the second sort of takeaway or inference I want to point you to is a set of topics that we call sort of meta topics. And this is where the question might be about anything, but it's framed in a way that's sort of about Dear Pandemic as a project. So this one right in the middle, which is a very common topic for us, we sort of call the "would you read this please" and have something to say. I think say is the big word here. So often the questions that come in the question box are kind of a "I heard this, would you vet it? I've seen this article, can you say something about it on the website?" And you can see the word trust appears in there as well. Versus the one over on the left with question and thank is really about people expressing gratitude for the project, which is of course enormously meaningful for us, but also an indication that identity and feeling like you're part of a community and feeling like you have sources of support to navigate, right, all the navigation that has to happen in the pandemic turns out to be really important for people, and we want to make sure that the project is responsive to that kind of question as well. Where next is a really important question for Dear Pandemic right now. This post on the left came out maybe in March or April. 2 of my colleagues actually coined this term, Pandexit. And we really thought we would maybe sort of close up shop in July 2021. We wouldn't need it anymore, or maybe we would pivot to covering other health topics. We really wanted to sort of take the brand. But that's not what happened. That's not where we are as of summer or fall. So we are going to continue to sort of be here for the community as long as they need us, while we do think about sort of future projects. But I think you'll be seeing more of our posts be either non-COVID topics or more on this sort of infodemiology. How do I become in general a good sort of information citizen and information consumer? Again, our platforms are all there. I hope they can be useful. Pass them on. We know there are a lot of sort of competitors for info, pandemic info. That's really not the case for Spanish language mediums. So if you have Spanish-speaking friends or relatives or communities, if Querida Pandemia can be of use, that would be wonderful. Happy to answer questions now, or feel free to email me if there's more you want to talk about. And I'll stop there.
There we go. Thank you. I've been having a lot of trouble today. Anyway, thank you so much for that wonderful talk. Really excited that now we have an option in Spanish, right, to sort of get that same information. Okay, let's see. I think we have a question. Let's see. But I mean, how has been sort of your experience, right? Like seeing this platform just kind of grow into what it is, and what would you say to people that are enthusiastic about initiatives like this, and really want to help and make a difference?
Couple responses. One, I've given this talk in some forums that are about like women in science or women in academia or women in medicine, and there are questions about both like, why is this an all-female endeavor? But also, why does it tend to be women who, regardless of their underlying discipline, sort of do the labor, and it really is labor, right, of science communication? And my enthusiasm for the project, I will say, is like tempered a bit by like, this is a side gig. Yeah, we're going to get some good papers out of it, and I will say for some of us, we really have. This has not been true for me, but some of us have had our research agenda really kind of moved. Like they were doing health policy, and now they're doing epidemiology. But the one thing I would say is just like be maybe more deliberate than we were able to be. We thought we were going to do this. I just saw the emails when I was recruiting my friends to help. They were actually Facebook messages, and I was like, "Hey, could you answer questions online for a couple of weeks?" I mean, that was the ask in mid-March 2020. All right, and now we have like a CEO, and it's just been hard, frankly, to kind of wedge it in. And we had questions too about, did you think about monetizing it or becoming a for-profit, or bringing yourself under an academic, umbrella of some kind? And these are just all questions that are really time-consuming. So in addition to wanting to communicate all the info, there was also all the work around managing it as a project. So that didn't answer your question, but some of the top of mind things just to sort of share with people.
No, thank you. This is a really good perspective. I mean, I feel are you recruiting more people? Like, can students at Penn sort of help and do what they can? I feel there's a lot of sort of not anxiety, but just like desire to do something that makes a difference.
Yeah. So that's a great question. Our CEO is currently Dr. Ashley Ritter, whom you saw on the second slide, who is here at Penn. And I will say we're still figuring out the best way to sort of process inbound interest. We've had some Penn MPH students and students at institutions where the other nerdy girls work do like field placements with us or internships. There's a big trade-off between having a huge editorial board and people not having to write very often versus having a much smaller group, but there's a little bit more like control and communication is easier. And there are not clear answers to that. So I don't even have a good answer for like what to do if you're interested in joining the crew.
Yeah. Thank you so much for your time.
Sure.
So now we will welcome our final two speakers today, Dr. Nawar Nasir, who is currently a Master of Public Health student but is a recent graduate of the Microbiology, Virology, and Parasitology, or MVP program here at Penn, as well as Katie Strellau, who is currently a PhD candidate in the MVP program as well. They both, as Penn trainees, recently launched and led this massive contact tracing initiative of over 100 volunteers as part of the Penn Health COVID-19 response team. And so with that, please join me in welcoming Noor and Katie. And thank you so much, Dr. Butenhag.
You're welcome.
All right, I think we can get started.
All right, I will go ahead and share the screen. So I just want to start by thanking Jay for that wonderful introduction. And you all hear us?
You are good to go. We can hear you and we can see your slides.
Wonderful. Okay, thank you so much. All right, so thank you so much, Jay, for that introduction. And we also just want to thank the organizers for inviting us to speak at this forum. We were really honored to be invited, and we're so excited to share our experiences with contact tracing. So between March and September of 2020, we helped develop, launch, and actually lead the COVID-19 contact tracing system for Penn Medicine. And at the time, we were both microbiology PhD students who participated in the public health certificate program, which was why we were asked to help lead this project. So as Jay mentioned, I am currently still a microbiology PhD student, but Noor recently graduated and is now pursuing her Master's in Public Health as Dr. Nasir. So with that, I want to hand the presentation off to Dr. Nasir to explain what contact tracing is and why it is important.
All right, thanks, Katie, and thank you all of you for joining. Before we dive in, we just want to give a shout out to the amazing leadership team that we worked with to lead this project. So Dr. Carolyn Canusio served as our faculty advisor. Rachel Fursten-Simon and Kirsten Claycomb were fellow co-leads on this team alongside Katie and I, and helped us with the day-to-day operations. And Dr. Austin Killeru was our clinical contact at Penn Medicine, and in addition to providing clinical guidance, also provided us with the names and contact information for patients who had tested positive, who we went on to contact trace. And all of our work was done in collaboration with Penn Center for Public Health Initiatives, Penn Medicine, and Philadelphia City Department of Public Health. And so we'll start off by just going over briefly what contact tracing is. You all have probably heard this term over the past 18 months, but just so we're all on the same page, this is what Katie and I mean when we talk about contact tracing, particularly in the context of COVID-19. So let's say we have this individual here who has a potential exposure, and a few days later, they start to present some symptoms. We'll call this day 0. And after they present symptoms, they get tested and they test positive for SARS-CoV-2. So they have COVID-19. This positive test result will trigger a phone call from contact tracers like us. And during this phone call, what we're really trying to identify are individuals that this person may have come into contact with in the days preceding symptoms and days following symptoms. So in the days preceding symptoms, this person may have been infected, but not necessarily aware of their infection, but they may have been exposing other individuals. And these other individuals, shown here in black, are the contacts of this red infected individual, who we'll call a case. We'll also ask this case about the places that they've been to, such as whether they've been to work, because then their work colleagues would serve as contacts as well. We also ask about the days following symptoms because they can continue to expose others even after symptoms clear until the infection resolves. And importantly, if they live with other people, then they'll likely have household contacts. So individuals that they live with are also potentially at risk of getting exposed. And so during this phone call, we let the infected individual know how they can isolate so they can protect those that they live with or they may interact with from getting infected. We then call the contacts and we notify them that they may have been potentially exposed. We don't tell them who they may have been exposed from. We maintain confidentiality. We just say, hey, you may have been exposed on this date, so here's what you can do to protect yourself. Now, at the time in 2020, the most we could really do was recommend precaution measures to minimize transmission. So this was usually just asking them to quarantine. Today, we can actually offer resources for testing, but this was not readily available last year. So that's how contact tracing works on an individual level, but how does it actually help prevent an outbreak at a societal level? So let's say we have this one individual here who tests positive, so they are our case, and we call them and they let us know of these 3 individuals that they have come into contact with. We then call these individuals and let them know that they may have been exposed and ask them to quarantine. And if these 3 individuals are able to properly quarantine, then the 3 individuals that they would have come into contact with in the back are protected. And we stop the chain of transmission and we've stopped the virus from spreading to the rest of the community. And so contact tracing really has benefits at the individual level as well as at the broader societal level. So at the individual level, we are letting people know that they may have been exposed. We're letting them know about the associated risk of exposure and what they can expect. So for example, if you get a call and you find out that you've been exposed and then a few days later you experience shortness of breath, then you can know that, oh, this might actually be a symptom of COVID-19 and not just me feeling tired. We also let individuals know about isolation and quarantine protocols, so how long to stay at home, how to minimize exposure to the people that live with them. And connected to this isolation and quarantine protocols is really point number 2, which is connecting individuals to other resources. So within the first couple of weeks of our contact tracing work, we realized that when we were telling people to stay home, we were getting responses like, if I stay home, who is going to go buy food for my kids? Or if I miss a day's work, I can't make rent. I can't pay my utility bills. And so we quickly realized that asking individuals to isolate and quarantine without providing them with the support system to meet some of their other social needs, such as food, housing, access to medication, or transportation to hospitals, was not going to work. And so we partnered with Penn's social needs response team, who then called these individuals and got them connected to resources to address the needs that they expressed. And finally, at the societal level, we are breaking the chain of transmission with contact tracing. So if we think back to the previous slide with the schematic, by alerting individuals of their potential exposure and asking them to quarantine, we are preventing the virus from finding more susceptible hosts and getting transmitted through the community. And so with that, I'll hand it over to Katie, who is going to talk about our direct experiences in leading the contact tracing initiative at Penn.
Excellent. Thank you so much, Noir. So we had several responsibilities involved in building this entire program. We just explained what contact tracing is, but we also had to learn about that ourselves. We had read about it in books, but we had never completely built a program like that before. So we had to really think about what is the workflow for contact tracing, not just theoretically, but also practically. And it's important to remember that this was very early on in the pandemic. At the time, there were no clear protocols for isolation and quarantine. And if there were any guidelines, they were frequently changing from week to week, if not day to day. I'm sure you all remember, we were originally told not to wear masks, and then we were told to wear masks, and wiping down groceries with disinfectant versus not wiping down groceries. And those were all things we needed to pay attention to so that we could adapt our guidance and make decisions on a day-to-day basis. We also were heavily involved in volunteer management. So Noir and I helped recruit a lot of the volunteers, and our volunteers, at the time, this was an entirely volunteer-led program. Everyone who was a contact tracer or was helping behind the scenes with with creating code to correctly assign specific cases and contacts to specific contact tracers. Everyone was doing that on their own time. So it really was important that we have this passionate group of people. A lot of our recruits were actually Penn students in nursing, social work, masters of public health, even biology. And we also had some trainees in these areas retirees in these areas who wanted to use their time to continue to help our community. After we recruited our volunteers, we also had to train them. And we had to train them not just only on how to know who to call and how to record the information during the call, but also how to do this compassionately. Because the people we're calling, they have either tested positive for COVID-19 or we are the ones letting them know that they were potentially exposed. And that's really scary and confusing, especially at the time in which we were doing this. So it's important to be respectful of that as we are calling all of these individuals. And we also had to adapt our trainings to provide additional information as necessary. So, for example, in the summer of 2020, in response to the murder of George Floyd, we decided to include training on trauma-informed interviewing to ensure that our volunteers were really cognizant and considerate of the complex, multi-layered traumas that our community was experiencing at the time, and frankly, is still experiencing now. So there were many layers. You don't just call someone and ask where they've been, you're calling a human being, and it's important to be respectful of that. And then we also supervised our volunteers. So after we had trained them, we sent them out into the field to make their calls. And as they were making their calls, we supported them with some of the really difficult situations that came up. And along the way, we had to continually develop our team and develop the project because there were some situations that came up. Day to day, Noir and I and Rachel and Kirsten, who we mentioned, were constantly on call responding to these urgent situations that arose. So, for example, sometimes we would call a contact to notify them of their potential exposure, but they had already been hospitalized or passed away. And for a contact tracer making that call, that can be very alarming to realize that you didn't call soon enough. And we also had to figure out ways to report unsafe work environments when our cases and contacts brought them to our attention. Who do we report that to? What is the protocol here? And sometimes the contacts would need an official letter to give to their supervisor at work so that they could actually get permission to isolate. And we had to build an entire workflow to obtain and provide that for them. And through all of this, Noir and I learned so much. This really was an all hands on deck approach. In order to meet the needs of our community, we worked with a very large interdisciplinary team, and we communicated not just with our interdisciplinary team, but also teams across the US and actually around the world. We communicated with teams in Boston and Ireland to learn about what they were doing and how we could adapt that to what we were doing. We also were very much emotionally on the front lines. Although we were leading this project from our own homes so that we could protect our community, we were definitely on the front lines. Every moment mattered, and we were working very long hours because we knew that every call that we made could make a difference. And eventually, Noir and I started to burn out a little bit, and we actually talk about our experience with leading this team and our mental health and all of the other effects of this project in this publication that we wrote for Science. And I think it really does a great job of summarizing how we were feeling. And now that this experience, for us at least, is over, it really has influenced our career trajectories. Obviously, Noir is pursuing her MPH, and I have adapted my microbiology thesis to be more epidemiology and public health related. So it really was a very impactful time. So Noir and I are talking about all of this from the perspective of what it was like in the spring and summer of 2020. But obviously, it's almost 2022, and we are still in a pandemic. So what can you all as audience members do to do your part? And the most important thing is to get vaccinated. Getting vaccinated is is just the number one thing that you can do to protect yourself and others. And even if you're vaccinated, another great thing that you can do is to wear a mask indoors. And for those of us who are joining this webcast from Philadelphia, Philadelphia has actually reinstated the mask mandate recently. So it's important that we continue to do this because it has been shown to be effective. And finally, I hope that our presentation today has convinced you that if a contact tracer calls you, please pick up and please participate and answer the questions. It can feel very personal and invading, but the thing is, we really do prioritize your confidentiality. And just like getting vaccinated and wearing a mask, helping with contact tracing is something that you can do to help other people even while staying at home. And with that, we would like to open the floor to questions. And thank you so much for listening. And thank you again to the organizers for inviting us and to all the other speakers. It's been a wonderful evening of listening to all of the incredible work going on.
Yeah, thank you so much, Katie and Nawar, for a really excellent talk and for your inspiring all of us, right? These are incredible efforts that you're doing alongside your doctoral training. So insane, but thank you so much. We have one question from the audience, and it says, okay, how has contact tracing and recommendations for contacts changed with the increasing vaccinations? Does being vaccinated affect your recommendations for the contacts?
Yeah, it does. So the guidance is constantly changing, and it also changes locality to locality based on community levels of transmission. But generally, the CDC recommends that if you are fully vaccinated and if you test positive, then you do the stay-home isolation as you would have if you were not vaccinated. If you are a contact and you have been exposed, then they recommend that you get tested within 3 to 5 days of exposure. And during that time, you wear a mask and you wear indoors until you confirm that you have tested negative. Or if you, for any reason, cannot get tested, then to wear a mask indoors for 14 days.
All right, thank you. So how can people volunteer to become part of this sort of network of contact tracers? And what's your advice for people who are like, just want to sign up, or maybe a little bit like shy or introverted, as most scientists are in some way, shape, or form?
I think that is an absolutely fantastic question, Jay. Unfortunately, because Nawar and I did wrap up our work on this project about a year ago, we have not been as involved. And actually, since we were involved in this project, there has been a shift from volunteer-based work to actually hired personnel. So it's really fantastic that now people who are spending all this time doing this are actually being compensated for their time. But my understanding is that there is actually a hiring process and everything. And right now, we could put anyone interested in being involved in the contact tracing efforts in touch with people who are currently leading that, but we do not have access to that right now.
Thank you so much, Katie. Hopefully there'll be some opportunities popping up soon. I really would like to thank all of our speakers again. They're gonna come up very soon. Thank you so much for distilling all this very complex information into something that is palatable for not just scientists, but also for the general public. We have a couple of remaining questions, so I will ask them. I know there's some that were already answered, but just for the people who are not like looking at the chat, right? I will start with a question for Allison that read, do you know anything about the audience, like their political views, etc.?
Yeah. So Facebook tells us a little bit about that, and the other platforms tell us even less. So we've actually done some surveying of our followers just through, not a random sample, just whoever wants to respond. And politically, the Facebook following tips Democratic/liberal, but not like all the way over. So we have people from across the spectrum, and I would say the average is sort of lean Democrat to moderate Democrat. And that's just for English language Facebook. It looks pretty different from what we can tell for creative pandemia. And Instagram's just a different audience generally, younger, more progressive.
Right, so another question for Allison. How do you stay sane in the face of active malicious disinformation, not to mention a wall of toxic misogyny?
So the toxic misogyny is real. We actually have troll patrol is one of the most important jobs on Facebook page, and we really try not to mute, real questions and real conversation and real dissent, but you cannot be an asshole on our site, and you especially can't be a toxic misogynistic male asshole. I will say an amazing source of strength and support and sanity is the nerdy girl community. And I said in the answer online that we actually, one of our channels on our Slack workspace is F the patriarchy. And we just use it as a safe space to vent, not just about the Dear Pandemic Project, but about our academic careers and being women in academia. So that's been just this incredible gift of the pandemic and Dear Pandemic, is having that community.
Wonderful. Thank you for that. I'm going to shift to César. So Tamer says, thank you for a fantastic talk. What are the steps for these tests to be commercially available?
Yeah, that's a great question, Tomer. So, I mean, we're now working on optimizing some of the parameters of the test, some of the technology. For example, stability. We want them to be stable, ideally not need refrigeration. So that's kind of from the research standpoint, that's what we're working on. And then obviously we need to, they need to be manufactured. So we would need to either partner with a company or license out the technology or create a startup to do the manufacturing piece, because you need to really mass manufacture them, right, to deploy them. So those would be, and then regulatory issues as well. They need to get approved for testing here in the US is through the FDA. So those are the three main, I would say the three main things that would need to be done to actually translate this technology from the lab all the way to people.
Right. And I think something that's very linked to this question that Katie asked, have you had any success in providing these tests to the low resource communities that they were designed for? And perhaps what are your plans after FDA approval, right?
Yeah, that's a great question. I mean, we're piloting one of the tests right now. That's all I can say at this point. And I have had conversations with people at Penn to maybe do pilot studies in developing countries, maybe next summer. But because of COVID, things are so unpredictable that we only had an initial conversation. So, I'm not sure what's going to happen. But that would be definitely one of our main objectives, is to, if we create a technology, if we develop something, if someday it's useful to people that need it the most, that would be like a dream come true, basically. So yeah.
All right, I think I'm just gonna maybe ask one final question to all of our wonderful speakers. If there's one thing or one piece of advice that you would give the general public as to how to live in these ever-changing times, how to find solace or peace, or how to understand a little bit better what is actually going on. Maybe that got too philosophical. My apologies.
I know mine right off the bat, so I can start. One thing I wish for sort of the public, maybe outside the scientific community, is that they gain some comfort with the scientific process, so that they recognize that the science will evolve. It evolved incredibly rapidly in this pandemic, and that's okay. That doesn't mean we goofed before. It doesn't mean that we're flip-flopping or don't know what we're talking about. We're actually following a scientific process and learning new things that sometimes replace prior knowledge. That's been such a source of stress and distress for the public, and I think if we sort of boosted scientific literacy and comfort with that process, we could reduce some of that distress.
I also have an answer on hand, which is which, by the way, Allison, I fully agree with that I have thought extensively about the need for improving scientific literacy. I hope that the public also sees the importance in public health and public health infrastructure. As someone who had to help rebuild that infrastructure, I would really love for us as a country to be prepared for the next time an epidemic occurs, because unfortunately there will be a next time, and hopefully it will not be as severe as this one, but we do need to be prepared, and we need to be doing surveillance and have programs in place ahead of time, and all of those things cost money. So I hope that we can continue to fund public health for an extensive amount of time after this has all wrapped up.
I think that I would like for people to really support basic science and understand that public health is for right now, but basic science is for it's a long-term thing, and there's 40 years of research that went into developing the vaccine in as fast as it did as well. And the other thing I want to sort of urge people is don't panic every time there's a new variant, because they're just another part of the virus life cycle, and they're really, I mean, the only thing to do about that is to get vaccinated and be careful. But I just wish the newspapers would not, or the journalists would not make it seem like each variant is something new to panic about. So try not to let that get under your skin.
I can speak to actually what Dr. Buttenheim presented on, which is sort of the infodemic that we are in. So I would really urge the public, as you are seeing tweets and posts on Facebook, to sort of think before you share, check your sources, check references like Dear Pandemic's website, and when you have friends that are coming to you with information, don't be afraid to engage in conversation with them. It does not have to be like a disaster at Thanksgiving dinner. It can just be a normal conversation between friends where you talk about the scientific process, as Dr. Buttenheim mentioned, and how it's constantly evolving and changing. And that's why some of the guidance that we get is constantly evolving.
Thank you so much for those wonderful words. I feel inspired just to use something a little bit. But I really wanted to thank each and every one of you for your time and your efforts. It really is a joy to watch, and a joy to see like how much each and every one of you has invested in this particular time in our lives, right? I think before we go, I would really like to thank all of the members of Penn INSPIRE who shared this, who brought ideas, who asked questions, who try to recruit all these wonderful panelists. It really is a wonderful community, specifically the Network and Partnership Development Team, which I lead, and was sort of the designers of this community forum. So Marissa Egan, Victor Vasquez Torres, Ben Truong, and more specifically, the MVP of this whole initiative is Ashley Brooke Robbins. So thank you so much. These are students that are doctoral science candidate students and are taking time away to put on events like this. So thank you all so much for your amazing efforts. To everyone in INSPIRE, thank you so much for your help to making this possible. So with that, I would like to bid people goodbye and stay safe and healthy, okay?
Thank you, everybody. Take care. Bye-bye.
Bye. Thanks.
Stay well.